So I'm going to talk to you today about HIV vaccine design, and I think it's highly relevant to the interest in germline genes and databases that Corey was talking about. And, in this work, uh, I feel badly that I haven't been able to attend an AIR meeting, but a lot of people on our team have been closely involved. Jordan Willis and Chris Cottrell both do a lot of stuff and insist on using AIR standards, and they're deeply integrated, and we collaborate a lot with Corey. We just had a paper come out with a lot of data from Corey's team, genotyping NHPs actually, and we collaborate a lot with Gunilla Hedestam in Stockholm, and she's been critical for our human clinical trials. So I really love this community, and I wish I could be there, and hopefully I'll be able to attend next time. So without further ado, let me just get to the story. I apologize if this is a little bit more vaccine and sort of HIV oriented, but hopefully you can see where kind of the germline concepts are being put to use, at least one example. So everyone is very familiar with the COVID vaccines and inducing neutralizing antibodies against the spike protein. HIV has a very similar spike protein that's shown here rotating around. It infects cells using a similar sort of conformational change mechanism. It binds to different receptors, obviously. And to make an HIV vaccine, we want to induce neutralizing antibodies against the spike. The problem, or one of the problems, is that the spike for HIV is much, much more variable than it is for SARS-2. And it's more densely glycosylated. The blue molecules flicking around here are glycans that make it difficult for antibodies to bind. The red positions are positions that are highly variable from one isolate to another, and the yellow positions are positions that you wouldn't call them highly variable, but they're not super conserved either. They're less than 80% conserved, I think, and the variation from one site to the other is not highly correlated. So it's really, really challenging to find places for antibodies to land on this thing and induce antibodies that will neutralize diverse strains. There's millions of strains worldwide. So it's a very hard vaccine problem, but it's one we do need to solve. We do know that broadly neutralizing antibodies against HIV can be produced by humans. They've been produced by a small percentage of people that get infected with HIV. Usually it takes years for them to develop. Sometimes it's a little bit faster. And there's been a lot of study of these. I don't have time to go through it, but I'm going to summarize in these bullets here that BNAbs, broadly neutralizing antibodies, neutralize diverse isolates, some of them up to 99% of HIV isolates, some less. They can protect against infection in non-human primates and in humans. We know in non-human primates that broadly neutralizing antibodies given in advance of a viral challenge can provide sterilizing immunity if there's a high enough concentration of the antibody present. Different classes of broadly neutralizing antibodies bind to different epitopes on the HIV spike. And because the HIV isolates are so diverse, we're pretty sure that an HIV vaccine will need to induce multiple classes of BNAbs in order to achieve sufficient neutralization, potency, and breadth to protect against diverse isolates circulating among humans. In our work, we are trying to develop germline targeting vaccines, and I'll explain what that means. We're trying to develop a vaccine that will induce multiple classes of broadly neutralizing antibodies, and we're going after BNAbs to these 4 different sites. And I'm going to focus today on the CD4 binding site because we have clinical data, but we have actually this year and very recently published a whole bunch of papers. In the last 3 weeks, we did publish 6 papers. And so I listed some of them here for the other epitopes. So if you're interested, you can go check them out. So what is required to induce protective broadly neutralizing antibodies? At the end of the day, we want plasma cells that secrete BNAbs at high levels and maintain high levels of IgG in the blood of vaccinated individuals, because when you get infected with HIV, there's no time to wait for a boost due to the infection. The infection over the next couple of days integrates into your genome, and there's no way to get it out using current technology, mostly. So we need plasma cells secreting bNAbs and having high levels of them. And we believe that we need to start from naive B cells that have the right genes that confer the potential for them to mature into broadly neutralizing antibodies. And those naive B cells that have those the right genetic properties, they are very rare. They have specific genetic features for each bNAb. So for each of those classes that I showed you, it has a specific structural mode of engagement, and that structural mode depends on specific human antibody genes. So not all recombination events can lead to a bNAb, a very small subset of them that have the right genes. But even for the naive B cells that have a BCR with the right genes, they won't all, that can turn into one class of bNAb, they won't all be identical. For all the precursors that do have potential to mature into a bNAb, those precursors are quite diverse. It's a diverse pool. You can imagine the junctional, the junctional positions in the CDRH3s are not, they're randomly generated every time, and many different junctions can contribute to a BCR that has potential to mature into a bNAb. And finally, these naive B cells are difficult to activate with HIV protein. So this is kind of the view, and how do we solve this problem? The strategy that we and many others in the field are pursuing is called germline targeting vaccine design. And it really keys off the priming immunogen and designing a priming immunogen that can find and activate those rare precursors and get them to mature at least a little bit and produce germinal center memory B cells that could be subsequently engaged by a different immunogen, more native-like immunogen, that would trigger the same broad pool of precursors that are hopefully primed by the priming immunogen, and basically a series of immunogens to walk the system along, increase the somatic hypermutation, keep going with all of the wide breadth of precursors that were triggered initially, and eventually produce plasma cells that secrete bNAbs. And today I'm really gonna focus on the priming step and the first boosting step, because we do have clinical data at the end. I'll take you a little bit further if we have time to get to mouse data. So the priming step is essential, and that's sort of very apropos for this meeting. We have to think about what are the germline precursors for each class of bNAb that we want to turn on. What does that diverse pool of precursors look like? We can define what kind of genes we think they need to have, but what are the areas of diversity that they're allowed to have? We need to design a priming immunogen that can consistently activate the large pool of naive B cells, and we have to be able to prime that pool of precursors across vaccine recipients. If we can't prime all people that receive the vaccine, the whole strategy isn't going to work. And so the priming immunogen needs appreciable affinity and avidity for diverse precursors, diverse human B cells that have the right genes. So today I'm going to focus on one project, the VRC01 project, that's been our lead project for germline targeting vaccine design. But check out those other papers that I showed you, because we've made a lot of progress in other areas. So VRC01 class bNAbs bind to the CD4 binding site. They directly compete with the human receptor that HIV spike binds to. And so the mechanism of sort of protection is pretty clear. In order to bind this way, these antibodies need to use a human VH1/2 heavy chain and a 5-amino acid L-CDR3 to engage this way. And at least in the precursor level, we found that they need specific alleles so far for our immunogen. They have to have STAR02 or STAR04, and all of the known bNAbs are derived actually from the STAR02 or STAR04 allele. But with those specific genetic constraints, there's many known bNAbs that fit into this class, VRC01 class, broadly neutralizing antibodies. They have very diverse heavy chain CDR3s, and very diverse light chains, V genes and J genes. So there's a very broad pool of these precursors, and we need a priming immunogen with appreciable affinity and avidity for diverse VRC01 class human naive precursors. So we spent a very long time, starting as soon as this antibody was discovered, devising this immunogen called EOD-GT860mer, germline targeting immunogen. It's a nanoparticle presenting 60 copies of an engineered GP120 outer domain. And this immunogen does have appreciable monovalent affinity and avidity for diverse VRC01 class human naive precursors. We've published many papers with many different colleagues, many collaborators, many mouse models showing that the immunogen can prime VRC01 class responses in stringent mouse models, attempting to recapitulate some of the aspects of human vaccination. And it can induce VRC01 class memory responses that can be boosted toward in the direction of BNAV development in mouse models. So the key question for us was, well, okay, we did a lot of work in mice and a lot of engineering, and we have lots of structures, but is it actually going to work in a human, which is where it needs to work? So we carried out this clinical trial, IAVI G001, and we published the data a little more than a year ago, and we showed proof of concept for germline targeting priming. This immunogen EOD-GT860mer, it induced VRC01 class precursors in 97% of vaccine recipients in this clinical trial. And I'm not going to go through. I'm going to tell a little bit of the story of this paper. That's right. Sometimes I do, and sometimes I don't. This is just showing, the frequency of participants that produced a VRC01 class response, low dose and high dose compared to placebo. But I wanted to go through two pieces of data that I think illustrate why the vaccine worked. So in this slide, I'm showing you that VRC01 class BCRs that were detected post-vaccination, they derived from many diverse precursors in each vaccine recipient. So we took the sequences of VRC01 class B cells, and Jordan performed hierarchical clustering of those to understand which sequences were related to each other and which were distinct, which were derived from unique precursors and which were derived from the same precursor. And so within each cluster, we believe the way we did the clustering, all the sequences in that cluster should have derived from the same naive precursor. And two different clusters, we believe, would derive from two different naive precursors. What I'm plotting here is for the low dose group, each symbol is from an individual in the trial. So this participant, we had across all the different time points assessed, we had about 75 VRC01 class BCR sequences recovered, 80 maybe. And in the clustering, it looks like that they clustered into around 30 different clusters. So that participant, we believe, at least from what we could detect, had VRC01 class responses deriving from 30 different precursors. And we plotted the number of clusters versus the number of sequences for all the participants in low dose and high dose. And the dashed line is 1-to-1, which would be every sequence corresponds to one cluster. And you can see the data is pretty close to 1-to-1. The median number of clusters per participant in the low dose group was 32, and it was 65 in the high dose. So just from what we could measure in like 100 million PBMCs at a few different time points, we can show that the responses were indeed derived from diverse precursors. And we think this resort accords with the fundamental hypothesis of germline targeting, namely that we need to trigger diverse precursors in every individual. I mean, we think this explains the capacity of EOD-GT860mer to prime consistent VRC01 class responses. And we think anyone who's trying to do germline targeting for HIV or anything else is going to need to have something like this. So to try to understand even more deeply how this worked and why it worked, we made a lot of antibodies and we measured their affinity for the immunogen using SPR. We made both VRC01 class antibodies taken from different time points, pre and post-vaccination, and we made competing antibodies that bind also to the CD4 binding site, but they don't have the genetic requirements for VRC01 class. So I'm going to show you data in blue for VRC01 class binding and in green for non-VRC01 class binding. We had historical data that we collected from Shane Crotty's lab. They sorted naive B cells and we made them and measured affinities, and we made VRC01 class precursors from naive B cells, not from people in this trial. We measured their affinities, and they had a median affinity of like 6 micromolar, and non-VRC01 class had around 20 micromolar. But as I mentioned, in this trial, we recovered VRC01 class B cells post-vaccination. And from those sequences, we could revert all the recognizable mutations in all the V and J genes, and we reverted in the middle of the D gene as well. So we could make inferred germline versions of the VRC01 class B cells that were detected post-vaccination. And that's what this data is here, the IGLs. We produced these. We measured their affinities for the immunogen. And you can see the VRC01 class inferred germline precursors have a median affinity of 100 nanomolar, whereas the competitor precursors were at, the median was at the limit of our detection, 100 micromolar or more. So the VRC01, to the best of our ability, the VRC01 class precursors that led to the observed post-vaccination responses in this trial, they started off with a massive 3 orders of magnitude affinity advantage against their competitors binding to the exact same epitope on the immunogen. And now I'm showing you to the right here data from VRC01 class and non-VRC01 class competitors at 4 weeks after the first vaccination, 8 weeks after the first vaccination. Everybody got a second immunization at week 8, and this is now, 2 weeks after the second vaccination, and now 8 weeks after the second vaccination. Across all, 1 vaccination or 2, all these time points, the VRC01 class responses maintain this massive affinity advantage. And so the design of the immunogen basically altered the competitive landscape in favor of the VRC01 class response. And we think that also obviously has to do with why the molecule worked. So I just wanted to show you that to kind of look under the hood a little bit, and these data are in the paper. Another aspect of that trial that I don't have time to go into, we just published in a collaboration with Nilla and Alan DeCamp this paper in MPJ vaccines, and this is more directly kind of in line with the germline targeting and allele characterization interest of the AIR community. And basically, this is again related to G002. Nilla and Martin made IgM libraries from each individual and did personalized IGHV12 genotyping. And then Alan performed a lot of mathematical analyses to help understand how the variation of VH12 genotype from one individual to another actually strongly influenced the magnitude of the VRC01 class response in the clinical trial. And the last sentence in the abstract says the results demonstrate the need to define population level immunoglobulin allelic variations when designing germline targeting immunogens right at the start of our effort and when evaluating them in clinical trials. So I encourage you to take a look at this. This took us a long time to get published. So I think the allele community doesn't have enough influence among reviewers, but maybe we should stand up for ourselves and push. And I would say also in this paper, we utterly failed to cite Corey's paper that he mentioned in the intro, and that was a total failing, and we should have, and his paper is a beautiful paper. So I showed you that for the VRC01 class project, we established proof of principle in humans that one can design immunogen and prime precursors and do it very consistently. So that's not inducing broadly neutralizing antibodies. That's just showing that this very first step is feasible, okay? That's this first kind of arrow bullet. What are the next major milestones for us that are trying to make an HIV vaccine? Well, part of this fundamental aspect of this strategy, aside from priming, is we need this concept of heterologous boosting to work. We need to be able to do that in humans. If you keep immunizing with the same artificial immunogen, you're not going to get broadly neutralizing antibodies. We know that experimentally and theoretically that makes sense. You got to change the immunogen to get all the SHM that you need and mature the immunogen and mature the BCRs toward becoming BNABs. So we need to show that heterologous boosting toward BNAB works in humans. And that concept is tested in another clinical trial that I'm going to show you now called IAVI G002. Beyond that, we need to go further and actually induce broadly neutralizing antibodies for one of those classes, at least one of those classes that I showed you in the beginning, binding to one particular epitope. And ultimately, to make a vaccine, as I mentioned, we need to demonstrate BNAb induction in humans from multiple classes simultaneously. So this is just showing you where we are sort of on the path toward hopefully making an HIV vaccine. You can appreciate that we've got to do as much as possible in human clinical trials, because we're talking about triggering human germline precursors and then walking them along toward making human BNAbs. We try to do as much as we can in very, very carefully engineered mouse models with a lot of very smart collaborators, B-cell biologists that can manipulate mice. And we try to do as many experiments as possible using non-human primates. But as you all can imagine, it's not the same thing as testing your vaccine in a human. And so we're going to have to do a number of clinical trials, and we need to be able to do them pretty quickly to develop a sequential vaccine like that. It took us a very long time to produce the proteins and get that first G001 clinical trial done. And we just don't have the time or the money to keep doing it using protein production. So we started collaborating with Moderna actually a long time ago, and now I'm employed by Moderna, which is a great place to work. And we're working with them to use Moderna's mRNA LNP technology to accelerate clinical development of HIV vaccines and testing. So I mentioned G002 and the need to test heterologous boosting, and we've done that clinical trial, and I'm going to show you the data. And the reason that I'm able to show you this data now, only a year and a half after we published the results of G001, is because we used RNA. It was much faster to manufacture and get it going, and much more affordable. So in this clinical trial, it had 4 groups. The first group was a repeat of G001. Everyone received EOD-GT8 60-mer at week 0 and week 8. Now it was delivered by RNA rather than adjuvanted protein. And so we need to know, does that work? We didn't know. Group 2 is you get the priming immunogen same, and now we're giving a heterologous boost immunogen that we know, and I don't have time to show you, but we just published in 2 papers in Science Translational Medicine and Science Immunology. This heterologous boost worked really well in mouse models. So does it work in humans? And in group 3, we gave 2 immunizations with the priming immunogen followed by an immunization with the heterologous boost, because we didn't know if 1 or 2 shots of the prime would be necessary. And our affinity data from G001 suggested we might need 2 shots of the prime. So we really didn't know. And the fourth one is immunizing with the boost by itself, that in our mouse models doesn't induce VRC1 class responses, but we needed to know that in humans, and we needed to understand what you get if you just immunize with this in humans. So I'm going to walk you through the data that we have now, and I think I have about 20 minutes left. Somebody should confirm that. If I should stop sooner than that, just let me know. So let's first look at the responses just to the priming immunogen. So I'm going to compare the results of mRNA vaccination in G002 to the data that we collected with protein plus adjuvant vaccination in G001. And what we're showing here is on the x-axis, it's weeks post-vaccination. Each symbol is a participant. G001 data is in blue, G002 data is in green. On the y-axis it's the frequency of VRC01 class B cells among IgG memory B cells in the PBMCs of each participant. So this is what we want to know. This is the fundamental readout. And you can see prior to vaccination, we sorted PBMCs from these individual trial participants, the volunteers, and there's hardly any VRC01 class detectable. 4 weeks after the first vaccination, this was the data from G001. It was super exciting. 0.01% is like 1 in 10,000 memory B cells are VRC01 class. It's a fantastic number. In G002, it was even higher. 8 weeks after that first vaccination, again, it works better. So you can see mRNA, 100 micrograms of mRNA, basically is giving us stronger VRC01 class responses than protein did, which was a good sign. Even more surprisingly, possibly, is when we looked at the VH1/2 genes of these VRC01 class responses. To make broadly neutralizing antibodies, we need to select for a lot of somatic hypermutation, and we know the kinds of somatic hypermutation we're looking for. Here, I'm plotting each symbol again as a participant in either G001 or G002. The data are the heavy chain V gene mutation, amino acid mutation in the VH12 gene. And each symbol is the median per participant at that time point. And so you can see at week 8, we actually have more mutation in G002 than we do in G001. And that holds up at week 16. And in fact, it's a lot more. It's about twice as much mutation. And actually, we don't currently know how to explain this result. It's correct. It's not a technical error. It's been validated. Several different teams have looked at the bioinformatics, and we have experimental controls as well. So this is a real result, and this is favorable for learning how to try to make an HIV vaccine where we need to select for lots of mutation, and we're very excited about this. As I said, we know, so that was talking about the percent mutation overall, but we also know we can also look for very specific mutations in the heavy chain that we know we want based on all the known BNAbs. And we wrote about this in the G001 paper. We called them key VRC1 class heavy chain mutations. And for BNAbs over here on the right, we can write down how many key VRC1 class heavy chain mutations they have. The median numbers is around 13, you can see. And so what we're plotting here is weeks, but every symbol is again a participant in G001 and G002. We're looking at the VRC1 class responses. On the y-axis, I'm looking at the 90th percentile number of key mutations in each individual at that time. So we're looking at like skimming the cream off the top. And you can see in G001, we were doing pretty well, but in G002, we're doing slightly better. So not only are we getting better overall mutation, but we're getting more mutations of exactly the ones we're looking for. We're still, even at week 24, we've got 4 median. So that's down here compared to BNAbs. We are not at the BNAb level yet, but still we believe we're on the way. So I mentioned that so that shows that RNA works really well for priming compared to adjuvanted protein. But the fundamental immunological concept that we wanted to test in G002 was, does the heterologous boost work? So let me walk you through that now. So this is a little bit more of a complicated slide, but I'm showing you on the y-axis, again, the frequency of VRC1 class memory B cells in the blood. They've all been sorted for binding to the core boost. So these VRC1 class B cells bind to the heterologous boost immunogen. That means they've acquired the ability to bind to the heterologous boost. It's more native-like than the prime, which is a good thing. And we're showing you data in gold for week 8 and purple for week 16, and then this blue color for week 24. And we're showing you the different groups. And I don't want to walk you through everything, but 2 shots of VOD is here. That's like everyone that got 2 immunizations with the priming immunogen. And it produced this frequency of VRC1 class responses that could bind to the core boost. And when you give a core heterologous boost, you get a similar frequency. So we didn't know what would happen. The core has lower affinity for naive precursors. There was a danger that the frequency would be lower. We thought maybe the frequency will also be higher. It turns out that it's about the same. And if you look at week 24, 2 shots of EOD, you get this number, and an EOD followed by a core, you get a similar number. So this was just kind of, okay, we're doing as well. We still have a very high frequency of VRC1 class B cells in the blood, but now we know they've advanced a little bit in maturation because they can bind to the heterologous boost, not only to the priming immunogen. What was very exciting for us was we got this response if you look in the EOD to core group. What I'm plotting now is the percent of participants that produced VRC1 class responses in the blood that were able to bind to the core boost. And if you look at the EOD to core recipients, 100% of the participants made this kind of response at week 16, and that was also true at week 24. So we were able to give a heterologous boost and have it select for the B cells we wanted that could bind to the boost extremely consistently. We also found a remarkable thing, that the heterologous boost was able to maintain very high immunofocusing. So bear with me for a second. On the left slide, what I'm showing you is on the y-axis, the percent of CD4 binding site specific B cells, VRC01 class B cells, that are, sorry, the percent of CD4 binding site specific B cells that are VRC01 class. So I mentioned that we're inducing VRC01 class and non-VRC01 class competitors. And so I'm showing you the percent of all of the epitope-specific B cells that are VRC01 class. Again, for G001 in blue, G002 in green. And you'll see, and we highlighted this a lot in the G001 paper, Legat et al. Science 2022, that in G001, 4 weeks after the prime, only like 20% of the epitope-specific B cells were VRC01 class. 75% were competitors. And that was maintained, that we never really got far above that number. Even after 2 shots at week 16 in G001, a minority of the epitope-specific B cells were the class we wanted. And that's also true when you deliver the immunogens by the EOD GT8 by RNA. Maybe we're doing a little bit better, but still we only get up to 50%. So even though the vaccination is working, it's inducing high frequencies of VRC01 class responses, it also, in some ways, they're not immunodominant, and we have to monitor the competitors like crazy. So the thing we were worried about is, when we give a heterologous boost, what's going to happen? Are we all of a sudden going to prime lots of new competitors? And will our heterologous boost possibly be good at boosting the competitors that were primed by the priming immunogen, and maybe not so good at boosting the on-target VRC01 class responses? And the answer is that the heterologous boost worked extremely well. So if you just, we don't have time to go through all the data, but this column has 2 shots of the priming immunogen. All the B cells in this graph were sorted with the core. And you can see that about a median of 80% of the epitope-specific B cells are VRC01 class. That's super high immunofocusing. But that is really what we were attempting to do in the design of our core. So the core immunogen was used to sort these B cells. It wasn't part of the vaccine in this group. It was used to sort the B cells. And what this is saying is, if you immunize with our EOD prime, then you can selectively pull out the VRC01 class responses using the core boost. So that just says, okay, the design of the boost immunogen was pretty good. But the question that we wanted to know is, if you give the core boost as an immunogen, now you expose the immune system to this different form of the CD4 binding site, can you maintain high immunofocusing? And the answer shown in this column here is yes. If you immunize with core, and then you sort with core, you still have very high immunofocusing. Now we're, 80% of the CD4 binding site specific responses are VRC01 class. So we're actually, heterologous boost is giving us an advantage. When you just prime with the germline targeting immunogen, you get tons of competitors. When you now can give a carefully designed heterologous boost, you actually can suppress the competitors to a pretty strong degree, which is exciting and I think is very important for the success of the overall sequential vaccination regimen. I mentioned about G001 that one of the reasons we think it was able to induce VRC01 class responses in 97% of the participants was that it could trigger diverse precursors. And this graph is showing the same thing, basically in a different way for G002. We did hierarchical clustering. We did it for the different time points and for the different vaccine regimens that were received. And you can see, for example, if you get EOD followed by core, each symbol is the fraction of VRC01 class B cells that are a unique clone for one individual. And so this bar is the median overall. So about 85% of all the VRC01 class responses in all the individuals after this heterologous boost, 85% of them derive from what we believe is a unique precursor. So we are clearly undersampling. These data come from, as I mentioned before, we're taking like 100 million PBMCs per person per time point, and the VRC team was doing sorting. Clearly, in that aliquot of the human PBMCs, there's just massive diversity of the VRC01 class responses, and we find that really exciting. And we think, as I mentioned before, that's really necessary. It's going to be not only important for priming, but we think in the concept of heterologous boost, if we want to mature these responses to bNAb, it's in our favor to have many, many different trajectories where many different clones or clusters are tracking along and trying to mature in their own way. So we're super excited about this data. I'm going to skip that one. I'm going to skip this one. I'm just going to tell you that we saw good mutations from the heterologous boost because I want to get to the affinity data. So for G001, we made antibodies for a lot of different B cells, and we measured their affinity for the antigens. What I'm showing you on this slide is affinity for the core boost, not for the prime, just for the core boost. And I'm showing you for the different regimens at different time points, week 8, week 16, week 24. But there's the two different colors correspond to something that we've never talked about before and we've never done before. So normally, when we're doing a manimal study or when we're doing, especially when we're doing a clinical trial, we get from one participant, we get a collection of VRC1 class responses from that person at that time point, and we want to make antibodies. We might, in G002, we have hundreds of VRC1 class antibodies from one person at one time point. We cannot make them all. We have to decide which ones to make to measure their affinities. So we just randomly, we use a random number generator and we randomly select them. And we usually make like 3 per person per time point, which is already an expensive thing to do. But we're trying to get a fair representation of what's the population of VRC1 class responses that time. That's the green data. It's labeled random. In our pre-specified plan for G002, we also said to ourselves, well, for heterologous boosting, yes, in the ideal world, we could just be tracking along the entire population of responses and get it all to mature, even sort of the least mutated, least mature like at each time point. Maybe we can bring them all along. But, maybe in the real world, we need to think about after each vaccination, we're going to get a distribution of clones, and some of them are going to have more of the good mutations than others. And maybe we need to think about the best clones being the ones that we really want to launch forward, and they have the highest potential to eventually reach bNAb. So before we started the trial, we said to ourselves, okay, we're going to make antibodies not only for randomly selected, but we called them cherry picked. We're going to, and I'm labeling them selected here. We counted the number of VRC1 class heavy mutations for every antibody in a person at a time point, and we took the top 2 or 3. I think it ended up being 3. So that's what selected is. And so what I'm showing you here is the green data and the blue data, random and selected for the different groups. You can see in the green data, the random data for the different 1 shot of EOD versus 2 shots of EOD versus 1 shot of EOD followed by heterologous boosts. In the green data, there's not much difference. So if you only made the random antibodies, you might think, well, it's not clear that the heterologous boost really did anything. And that holds up for 2 shots of wait to week 24, 2 shots of EOD versus EOD followed by core. The median response is the same. Yeah, there's a few more that have higher affinities. Lower values are better here. But still, you would think that, it'd be hard to tell that the heterologous boost has done anything. But look at the blue data. If you look at the best responses, you see orders of magnitude improvement between random and selected, first of all, in the same group, but also we compare 2 shots of the prime versus 1 shot of the prime followed by a heterologous boost. There's, again, 3 orders of magnitude, 4 orders of magnitude difference. So clearly the heterologous boost did do something, and it's most detectable when you look at the best responses. And we're excited about this finding because we sort of thought this might be true, but it also focuses our attention when we're doing these clinical studies on the right population of B cells. So really quickly, in order to make a VRC1 class vaccine, we need to induce antibodies, VRC1 class antibodies that can accommodate the N276 glycan, which is the glycosylation site of glycan right at the edge or part of the CD4 binding site. And our immunogens currently don't have that in there. In our vaccination plan, we introduced the glycan later in the series. But what we're showing you here is the selected antibodies from the heterologous boost group actually have some affinity for the core immunogen with the N276 glycan added back. So increased heavy chain mutation is allowing them to take the affinity hit incurred by the glycan being present. These same selected antibodies. I'm showing you now SPR data for the selected antibodies only from the different regimens, not showing you the random antibodies anymore, just the selected. But now we're asking, how well do the antibodies bind to different HIV envelope trimers from different isolates? All of them, the only change we've made is to remove the N276 glycan, because we know that that kills the binding. And you can see that the heterologous boost gave you antibodies that bind much better to these heterologous trimers than homologous boost. So again, direct evidence that the heterologous boost was really working and driving us in the right direction. And we've actually carried out neutralization assays with these monoclonals. And we obviously don't have time to go through all this data, but these are the antibodies from week 16 and week 24 that came after one shot of EOD followed by one shot of core. And they don't neutralize wild type isolates that contain the N276 glycan at all. But you can see from the colors that they do a pretty good job of neutralizing multiple different isolates that lack the 276 glycan. And at week 16, 54% of the antibodies neutralize more than one different isolate. So this is getting closer to BNAB. We aren't at BNAB yet, but a direct functional readout showing in humans monoclonals of the exact B cell class we're trying to turn on are walking in the right direction. So we're really excited about this data. And my last data slide is to come back to those selected responses. So I showed you that if you just look randomly at all the VRC01 class responses, you can't really see a great affinity advantage. But if you look at the selected ones, you see an affinity advantage. They can bind to trimers. They can neutralize pseudoviruses lacking the glycan. So we said to ourselves, okay, well, if that sort of, we called it an elite population of VRC01 class responses, if that elite population that actually has 5 or more key VRC01 class residues, those are the ones that have the neutralizing activity. If that elite population is really the one that we want to target for future, for further maturation toward BNAB, we need to know how common it is. I mean, if it only happened in a few individuals, it's just a curiosity. It's not meaningful. And so what I'm showing you now in this slide are 3 different panels of data characterizing how common are the elite responses that have 5 or more key VRC01 class residues in the heavy chain. And so what percent of participants did we detect those elite responses in? EOD to core at week 16, 80%. EOD to core at week 24, 95%. So we do see them basically in almost everybody. How frequent are these elite responses in the blood of the positive responders? EOD to core, the median frequency is 1 in 28,000 memory B cells. 1 in 28,000 memory B cells in each individual is an elite VRC01 class B cell. That is a remarkable number, and we believe that is high enough to be ready for the next heterologous boost to further drive that along. So we're really excited about that. And finally, you might be concerned that, yes, we see these elite responses and they can neutralize and they can bind tightly, and yes, they're common, but maybe you've gone through a clonal bottleneck. Maybe you've just got a few clones in each individual that can do that job. And if you're going to try to further mature them, you're going to run into trouble. But that is actually not true also. So we're showing the percent unique clones here. We did hierarchical clustering. EOD to core week 16, 85% of these elite VRC01 class B cells represent a unique clone in the data that we have. So basically, you couldn't ask, I don't think, for more than this for this stage of the clinical trial in terms of saying we think we're ready for future, a next heterologous boost to drive the system forward toward making BNABs. So conclusions from these studies: mRNA LNP delivery of the EOD to core regimen generated strong and consistent VRC01 class responses with increased maturation toward BNAB development. The data demonstrated clinical proof of principle for heterologous boosting of a pre-specified B cell class. mRNA LNP delivery of self-assembling nanoparticles appears promising for vaccines in general. I think this is the first human data from mRNA delivery of self-assembling nanoparticles. You may have heard, I didn't have time to show, we do have some dermatologic adverse events in G002 that we didn't see in G001. and we need to understand them better and understand potential for mitigating them. One thing we're doing is just coming back with a lower dose. The next step for this clinical program is to test lower doses to determine if we can decrease skin adverse events while retaining sufficient BNAB precursor priming and boosting. And I would say the strong responses in G002 seem to allow for reducing immunogenicity and still achieving BNAB priming at the level of G001, which was already exciting. And I don't think I'll have time to show you today. I was hoping I would, but in preclinical experiments, we are testing more advanced sequential boosting regimens going beyond just this core boost, working our way toward BNAB induction in stringent mouse models. And hopefully, you know, a year from now or some months from now, we'll be able to report out on that. Obviously, this work comes with many, many partners. I have to thank all of them. Scripps, Moderna, Gates Foundation, NIH, the CHOB, all of our collaborators. I mentioned the vaccine. All that data from G001 was from B-cell sorting at the Vaccine Research Center and at the Fred Hutch. And in G002, the Fred Hutch, and then at G002 is all done at the Vaccine Research Center, so that's fantastic. Obviously, lots of people contributed to these studies. I just want to call out the team at the VRC, Sarah Andrews, Madhu, and Weiwei for doing the B-cell sorting and sequencing, and Jordan Willis Troy for doing the bioinformatics and making all the gorgeous graphs I just showed you, analyzing the responses, and Chris presided over the SPR data and the neutralization assays. And I will stop there because I think my time is up.
Okay, Bill, thanks so much. That was amazing. Are you okay to take a few questions?
Oh yeah, sure. No
you
problem.
have many more than a few, but I don't know that we have time for all of them. So
okay,
but I will do my best to try to.
Well, if we don't get to them, people can email me, and I'm happy to correspond.
So I'll start with the general one. So why is heterologous? I can't say that word ever. Boosting beneficial for SHM?
Good question. So you can imagine if you expose the immune system to the same antigen, say 2 or 3 times, the way the immune system works, B cells see the antigen, they go into a germinal center, they randomly gain, they go between the light and the dark zone, they randomly gain mutations, and then they're tested for whether the mutation improved the affinity, whether the B cell can acquire more T help. And so in the operation of the GC, it ends up producing B cells with higher and higher affinity for the antigen that was delivered. And you can imagine that because we're all alive and we all have survived many different infections, that system works pretty well. So it only takes you a couple of exposures until you have really high affinity antibodies against whatever the antigen was. And once you get to a certain affinity, the way the GC works, it can't really select for higher affinity, and you get stuck. So if you immunize with so that's one point. And then the other point that you need to understand is to make a vaccine against SARS-2, we could just immunize with the native spike. And we get high affinity antibodies to the native spike, and they neutralize the native spike. And because there aren't that many different variants of the native spike, at any one time, you can have one SARS-2 vaccine, neutralize everything, and you're good. Seasonally, we update the vaccine because the spike is mutating. We need to make a new vaccine, but still, you can update. So you can get antibodies that are high affinity against the vaccine, they'll neutralize everything. For HIV, you cannot do that. If you give one trimer of HIV, you might get autologous neutralizing antibodies against that strain, but they won't neutralize any of the other million strains that are present around the globe. And so we cannot start with a native trimer immunogen. We have to start with a germline targeting immunogen that's highly engineered to have affinity for the precursors of broadly neutralizing antibodies. So the epitopes that we expose in the prime don't look like the native pathogen. So if we reach super high affinity by giving 2 or 3 shots of the prime, if we reach super high affinity antibody to the priming immunogen, that'll be great if the virus was presenting the priming immunogen, but it's not. those antibodies won't neutralize HIV and they won't help us for a vaccine. So it's those two issues. We have to prime with a very engineered immunogen that's pretty different from the native. We have to give sequentially antigens that are more and more successively like the native antigen until finally at the end, the final boost will be probably a cocktail of native trimers. And along the way, every time the GC responds, we gain affinity to the antigen we just gave. So it's kind of a directional process. I hope that answered the question.
Yeah, no, that was great. Thanks. Another question. Can you describe how the cherries were selected for the affinity test? And would you expect similar results if you just selected the most mutated antibody sequences?
It's a good question. The way we selected the cherry-picked antibodies, and we're not calling that we're not going to call them in our paper, but as I said, we have a list that we've published. We have a list of VRC01 class key heavy chain residues. And I call them residues, not mutations, because most of them are mutations away from germline, but 4 of them are germline encoded. And so we count them. We want to make sure we don't lose them. So the way we did it was for each set of antibodies from each person at each time point, we count the number of key VRC01 class heavy chain residues and we rank them. And then we took the best one, the second best one, and the third best one. And if they were tied, we randomly selected among the ones that were tied for the best. So it was totally algorithmic. And yes, there is the questioner also said, if you had just taken the most mutated, would that have also done the job? There is a correlation between number of key VRC01 class residues and percent mutation, but I don't think it's perfect. And so I think it probably would have done some of the job, but may not as perfectly, as we showed you. In cases where, maybe the questioner is thinking we may have another case where we don't know enough and we may not know what all the key mutations are, and maybe we should just focus on the most mutated B cells as a proxy, and that is a good idea, and we are doing that. Although most of the time, we try to understand our system well enough at the structural level such that we can, we can sort of rank in terms of how good they are. But in some cases, we can't do that.
Okay, next question, if you're willing. Do you think the nanoparticle is important compared to, for example, just the same antigen anchored on the cell surface, similar to the COVID vaccine?
Yeah, also a really good question. We've actually been trying to set up a formal test of that for a long time, and we can make, I don't know the answer, to be direct. We can make membrane anchored EOD-GTA perfectly well. Where we've gotten hung up is in matching the CD4 T help in that construct with the lumazine synthase nanoparticle, because the underlying nanoparticle, which in this case is lumazine synthase, to which EOD is fused, that underlying nanoparticle has a lot of T helper epitopes that work super well in mice and in humans. And we have a paper, Kristen Cohn et al. From last year that demonstrated that in humans. And so we've avoided doing the experiment in mice until we think we have a fair comparison of T help, because otherwise the answer is kind of ambiguous. I suspect that it'll be close, but the nanoparticles still might do better, but I don't know for sure.
Great. Okay, one more, and then we'll let you go. So does the boosting strategy have any risks in terms of inducing enough selection pressure on the virus to induce viral escape? Tried to paraphrase that, and I might have made it
That
worse.
is a good question. And obviously all good questions. So the antibodies that we're inducing while we're doing the sequential vaccination strategy, they actually don't neutralize circulating viruses, and they don't bind with any appreciable affinity or detectable affinity to trimers corresponding to circulating viruses. So in that way, they don't put any pressure. Once we start getting to neutralizing antibodies, if we elicit kind of low levels of neutralizing antibodies, then you could imagine the potential for a person to be vaccinated, have sort of low levels of antibodies, get exposed to the virus, and now there's a tug of war, and maybe the virus will escape the response. And that's one reason why what we're focused on is inducing multiple different classes of broadly neutralizing antibodies so that it's really hard to escape the sum total of all the responses, and maintaining really high titers so that it's really hard for a virus to actually infect someone that's been vaccinated. And that thing about, really high titers, we have good information from the AMP trial about just how high the titers need to be for 90% protection in humans, and it's pretty high. And we're still, right now, we're focused on getting the specificity, namely the breadth and the epitope specificity. And in parallel, we're working with people and thinking about, all right, once we get that specificity loaded into memory, what are the best strategies for maximizing the titers? And that, the field is still working on that as a whole, I would say.
Well, great. Let's give Dr. Schief another round of applause, and we'll let him go.
Thanks so much. I wish I could be there. Have a good rest of your meeting, you guys.
Next time.